Myself, Dr. A S VIJAYAKUAMR, Assistant Professor, Department of Biochemistry, JJ College of Arts and Science, Pudukkottai. I am going to take about lipoproteins. Lipids introduction. Lipids are either compounds that yield fatty acids when hydrolyzed or complex alcohols that can combine with the fatty acids to form esters. Lipids are carried in the bloodstream by complexes known as lipoproteins. This is because these lipids are not soluble in the plasma water. Lipoproteins. In order to be transported in blood, lipids must combine with water-soluble compounds such as phospholipids and proteins. Transport lipid to sites of utilization or storage. Composed of variable amounts of lipid and functional apoproteins. Plasma lipid levels represent balance between absorption, synthesis and utilization clearance. Change in blood lipid and lipoprotein components associated with atherosclerosis, thickening of the inner layer of the arterial wall, lipid storage disease. These are the classification of lipoproteins. First one is chylomicrons, and another one is VLDL and LDL and HDL and lipoproteins, apolipoproteins. Chylomicrons produced by the intestine transport dietary lipid from intestine to fat muscle cells. The largest lipoproteins are chylomicrons, mostly lipid, almost 90% triglyceride, 1% protein, mainly APO, B48 and C2 and E. Lipid poor remnants removed by liver, APO, B bar E receptors. And VLDL produced by the liver, transport liver synthesis of fat to fat muscle cells, mainly lipid, 55% triglycerides, 10% protein and APO B100 C2E. Remnants termed intermediate density lipoproteins cleared by liver, ApoBE receptors. And LDL produced from IDL with the loss of ApoC2. Transport cholesterol to tissue taken up by ApoB receptors and cholesterol released in liver decreases cholesterol synthesis, decreases ApoB receptors. Alternate pathway for uptake is scavenger receptors, mainly on macrophages. Leads to the formation of foam cells. Does not inhibit cholesterol synthesis. Modifications to LDL increases activity of this pathway. Prothrogenic, better marker for CHD risk. And HDL, the smallest and most dense lipoprotein, synthesized by liver and intestine. Mainly proteins are ApoA1 and A2. Lipid cholesterol phospholipids involved in reverse transport of cholesterol from tissue to liver. This is apolipoproteins. This is the structure of apolipoproteins. The protein composition differs from one lipoprotein class to another, and the protein constituents are called apolipoproteins. Functions of apolipoproteins: activate enzymes involved in lipid metabolisms, lecithin, cholesterol acetyltransferase, lipoprotein lipase, maintain structural integrity of lipid protein complex, delivery of lipids to cells via recognition of cell surface receptors. Apolipoproteins contents of LPS. These are the reference of lipoproteins. Thank you.